Hello, everyone. Thank you so much for coming. We are gonna start now. First of all, thank you so much for coming for MEXPOP Gen 4. We are delighted to host it here in Mexico City at the Faculty of Science. First, some remarks on safety. This is because I studied in England, you see. So the first half of the auditorium has to evacuate in by the lower stairs here, and the upper half goes to the main entrance. As you know, Mexico City is a seismic place, so in case of earthquake or seismic alarm, please follow those instructions. Now that I told you how not to die, I would like to highlight why we choose this beautiful picture to illustrate MEXPOP Gen 4. It was first because it represents the Pedregal de San Angel, which is the volcanic soil ecosystem where UNAM is, but also because it shows a wide range of organisms, which include plants and animals and bacteria. You cannot see them. And that's because we wanted MEXPOP Gen 4 to include not only humans, as in previous editions, but to have a wider representation of the study systems population geneticists of Mexico work with. So this is a word cloud with the study systems of this MEXPOP Gen. There's a lot of work in humans, which is great, but also, if you see, there are many others. And if you group them, you will see that we have a wide range of works, especially in plants and non-human animals, which I think it will be great to have a lot of diversity, and also we will have methodological presentations. Some logistic remarks. We will try hard to keep on time, so please tight to your allocated time. At lunchtime, there will be some provided for the ones who registered. And the idea of this is that we can be more social and interact and have further discussions. And also we have a posters and posters section. So please, if you're presenting a poster, grab your basket of dessert, Mexican desserts, and lure people into your poster with it. I would like to acknowledge all our sponsors who made this Congress possible, but especially the real heroes of this, Maria, who is the intellectual mind behind MEXPOP Gen, and especially Dalia and Salia and the rest of the students who helped us. With that said, welcome, we are ready, and let's start. So in our morning session, our first keynote talk will be by Diego Cortez. ¿Puedes ayudar? So Diego is an associate professor at the System Biology and Synthetic Biology Laboratory in Genomic Sciences here at UNAM. And today he will be telling us all about genomics of sexuality in vertebrates. So thank you so much for coming.
All right, thank you very much. First, I want to thank a lot Maria Avila for the invitation, and Andres because they are very close friends. So I'm going to talk today about a little bit of a genomics of sexuality. So for many, many years, I worked with microbes, and then one day I decided to change my subject of research, and I realized that there were funnier things to work on. So that's when I started working on sex. So particularly, I started working on sex chromosomes. So I'm going to talk a little bit about sex chromosomes in general, and then I'm going to talk about the main topics that we are investigating in our group. So the idea is that we have sex chromosomes. Many species invertebrates have sex chromosomes. In our case, so placental mammals, we have what we call an XY system that consists of females having 2 X chromosomes and males having 1 X chromosome and 1 Y chromosome. And it's actually this Y chromosome that makes the whole difference. That is, placental mammals, they are all born as females, and for many, many weeks they develop both males and females with the same program, until we have one change. This change comes from one specific gene, in our case, that is called the SRY gene, that is located on the Y chromosome. This gene gets activated at a specific time point, and at that time point it changes the program, and instead of developing ovaries and then becoming a female, we develop testes and then we become males. So this is the whole secret. If we don't have SRY, if SRY is not activated at the precise timing, then we will continue with the default program, and that is, we will become females. So the idea is having sex chromosome is great for a population. Why is that? Well, I mean, compared to having determination by the environment, like temperature or photo periods and things like that. So sex chromosomes are very useful because we actually get, at the population level, 50% males, 50% females every single generation, independently of having climatic changes and starvations and things like that. So this is the good side of having sex chromosomes. And we also have a bad side of having sex chromosomes. And that is, the moment we have this master regulatory gene, in our case the SRY gene, that appears in the population, then because it's so important to have this gene in our population, then the whole chromosome gets isolated from the rest of the genome. So we stop recombination at the SRY locus, and then this arrest of recombination spreads around the whole Y chromosome. So at some point, the whole Y chromosome stops recombination. And the recombination is very important because it actually fixes lots of deletions that we have in our genome. So with time, at the moment that we have SRY, we stop recombination, and then little by little, this Y chromosome started to lose genetic information. And at some point, we have, what we have now actually in our case, what we call degenerated sex chromosomes. So the Y chromosome is actually a degenerated X chromosome that only have like around 2% of the original gene content that used to have when it was an autosome, or it used to be like an X chromosome. So this is very important for the rest of the talk. So remember this 2% because it's going to be very important later. So this idea is not new, that we have a problem between having 2 X chromosomes in females and having 1 X and 1 chromosome in males. So this is something that already in the 20th century Susumu Ono realized by looking at karyotypes, and then he actually realized that we have a big problem. And the problem is that we have unbalanced sex expression levels between males and females, because either females have too many genes because they have 2 full X chromosomes, or males have too few genes because they only have 1 X chromosome and then 1 tiny Y chromosome. So the idea is, how do we balance expression levels between the 2 sexes? So let's take a break for a moment and then realize that we are all very beautiful. So vertebrates are very beautiful. So here a nice picture of a quetzal flying in Chiapas. But this is a lie. Why is this a lie? Because we, as Darwin realized many years ago, we are tricked by sexual selection. So we look beautiful, that is, we actually look like a nice red car. But in fact, in the inside, at the genomic level, and this is what I'm going to develop a little bit later, we look like this. So this is actually how we look at the genomic level, although sexual selection makes us look beautiful from the outside, phenotypically. And this is particularly true for sex chromosomes, and let's see why. So at the beginning, before we had sex chromosomes, we had like a pair of normal autosomes, and then these autosomes, well, everything was balanced, right? We were very happy because we had the same expression level for these 2 chromosomes in females and then these 2 chromosomes in males. And then we had, at some point, the origin of the master regulatory gene for sex determination, and then one of the chromosomes decayed. So now we have a Y chromosome that decayed, and then we have a severe unbalance in our system that we need to restore. And this was the main question that Susumu Ono asked in the beginning of the 20th century. So what do? We patch our system. How do we patch this system in this case? So in placental mammals, we realized that we need to inactivate one X chromosome. So we have a huge complicated system based on CIST, which is a long non-coding RNA, and inactivates the whole X chromosome, specifically in females. So CIST is only expressed in the Y chromosome, in the X chromosome that's going to be inactivated. So we inactivate one X chromosome, and then we almost balance the system. That is, we have one fully X chromosome activated in female, and then one X chromosome activated in males. But then remember the 2% that I told you that was on the Y chromosome, because this is true, we balance the sex chromosomes, but for 98% of the genes. But we have 2% of genes that are on the Y chromosome that are shared between the Y and the X chromosome. Now we need to rebalance this whole. So now I spend a lot of time, evolution, mechanisms, proteins, inactivating one X chromosome. Now I need to somehow restore what I call the second patch of our system. So we need to repatch our system and then do something about these 2% of genes. So in this case, we are gonna make a new system that is escaping. So we have a second mechanism that makes specifically for these genes to escape X inactivation. So first I inactivate the X chromosome, and then I make these few genes to escape X inactivation. So this is like a very long story, so people have been working on this subject for the last 2 or 3 decades. So what we're doing actually in the lab is that we realize we also almost balance this system. And why is that? Because the Y chromosome has not only lost genes over the course of evolution, but it also has lost gene expression. So when we compare the gene expression that the X chromosomes have compared to the ancestral gene expression, and then we see the expression of the Y chromosome, we actually realize that the Y chromosome has lost almost 3-fold the original gene expression of these genes. So the Y chromosome is losing gene expression, basically because we are accumulating slightly deleterious mutation at the promoted regions. So with time, the Y chromosome is producing less and less transcripts that will produce less and less proteins. So this is also an imbalance in our equation, right? So what we are trying to understand in the lab is how do we balance this equation back? So one idea that we had is that we need to, so some of these genes, that is something very specific in vertebrates, can escape some chromosomes by retrotransposition. So you can get transcription that is reverse transcribed by a reverse transcriptase and then relocated to a different chromosome. And if you do this several times, then you have extra genes on different chromosomes that are actually producing extra mRNA and are producing extra protein for the system. So this is what we've been investigating with several students of the LCG. So there is one there is Laura there. But also, so this will be like a small, like a more discrete patch for our system. And then we realized that we actually, after investigating lots of retrogenes in around 20 different mammals, this is like the slide that makes the summary of all these results. We realized that these retrogenes actually might be the key to restore balance to the whole system. So if we add the expression of retrogenes to the whole XY problem of dosage, we regain what we call a full dosage compensation. So this is not true for all species, this is not true for all the genes, but like the general picture seems to be this one. So after all these patches, right? So somehow the system works, but genetically we have a lot of problems, and then we realized with time that many of these problems have an impact on the sex that is carrying these degenerated sex chromosomes, and in almost all amniotes, The sex that carries the degenerated sex chromosomes is dying before the sex that is not carrying the sex chromosomes. So in our case, that's probably why females are living longer than males. But in birds that have the opposite sex system, actually males are living longer than females. But that's a different story that I'm not gonna talk about today. So in exploring this dosage compensation in other different systems, now we moved to more interesting species that will be reptiles. Reptiles are very interesting because they have tons of different sex determination systems. So they have sex chromosomes, but they also have microchromosomes, and they also have environmental sex determination by temperature. So we chose to work with the model organism, in this case the green lizard, Anolis carolinensis. And then we did the whole process that we used to do. So we sequenced the genome, we sequenced the transcriptome, and then we identified the sex chromosomes and the genes located on the sex chromosome, specifically on the Y chromosome that is actually very complicated. So we found actually a very degenerated Y chromosome with only 7 genes. Only 4 of these genes are shared between Anolis carolinensis and the iguanas. So we have a very old system of 160 million years old with very few genes. So ideal for a system that needs to be balanced, right? Because females will also have 2 X chromosomes, and then males will have 1 X chromosome and 1 highly degenerated Y chromosome. So we tried to understand how this system was balanced, specifically in this case. So we realized that Anolis was a perfect system based on what Susumu Ono predicted, and that is you have a perfect compensation between males and females. There is no difference between the X expression when you have 2 X's in females and when 1 X in males. So there is definitely a compensation mechanism. And then we realized that when we compare to the estimated ancestral expression, we also have perfect compensation. So in humans, we inactivated 1 X chromosome, but we didn't regain the ancestral expression. We stayed with 1 X chromosome activated. In this case, we're actually having the expression of 2 X chromosomes all the time. There is something happening, and we thought that the idea was the X chromosome in males is upregulated. How do we go to this idea? Because we did a little bit of gene expression analysis, and then we realized that we had 1 gene that is highly expressed in male organs compared to female organs. And this gene is this one, this APPB1, that is involved in the acetylation machinery. So acetylation is very important for gene regulation. When you have your chromosome is highly acetylated, then you increase. So when you have the chromosome that is highly acetylated, you increase the level of transcription because it opens up the nucleosomes. And then the transcription factors can locate the regions easier. So if you increase the acetylation, you increase gene expression. And so that's exactly what we did. So we did the experiments. So this is like a cartoon of the experiments. So we have the female with 2 fully expressed X chromosomes, and then males with this huge imbalance. And then when we realized we need a huge, massive patch for the system, and then this patch comes from the acetylation, a specific acetylation of the X chromosome in males. So the X chromosome, we don't know the system. We have some candidates, and we actually now trying to do some experiments with people from Langevio and Timberstaff, so with Dr. Kasha and Dr. Selene, to try to understand if we have a long non-coin RNA also triggering this acetylation on the X chromosome. So we have a full acetylation of the X chromosome, and that makes the whole X chromosome in males to regain the double of the expression and restore balance between the 2 sexes of the system. So this is a perfect balance equation. And this is very surprising because it's actually exactly the same mechanism that we see in very distant organisms like Drosophila. So the Drosophila has the same problem, 1 Y degenerated, and then they found exactly the same system by acetylating the X chromosome in males. So this tells you that we actually have very few options to solve the problem. In this case, we're actually getting the same solution from completely distant organisms. So I'm glad that Agustin is here because I want to conclude this part with a little bit of a text that he wrote for when we published the paper, and it says, Susumu Ono died on January 13, 2000, although he received great recognition for his work in the University of in the United States, Europe, and also by the Emperor of Japan. surely one of his favorites would have been to know that his predictions regarding the sex chromosome were fulfilled by a lizard, which is actually very beautiful. So the last thing that I wanna talk to you about today is what do we do when we spend a lot of energy and time, lots of evolution to get this perfect system. So it's perfectly balanced between males and females. And then suddenly, what do we do when everything works? Well, we start all over again. That's like the magic of biology. So we have one lizard. This is a very Mexican lizard that is called Jesus lizard because he can run on water. So it's good that it's a Mexican lizard because we can actually go on the field and then capture this lizard and bring them back to the lab and try to work with this species. So this species is the one I showed you is Basiliscus vittatus, that is part of the Corytophanidae family. So Anolis carolinensis are model organisms down here, in the Dactylidae family. And then all of these species, all of the families here in black, they share the same XY system that I told you about, with a X-compensated system and then a highly degenerated Y chromosome. And then suddenly we realize that this family looks a little bit strange. So it seems like the X chromosome in males and females is always in 2 copies, which is kind of strange because the males should only have 1 X copy, right? So we started to understand, right? So we sequenced the genomes of these lizards, and then we actually showed that regardless of the chromosome, we have the same coverage for all of the main chromosomes. So for males and females, we have 2 copies of the sex chromosomes, of the same chromosomes, including the canonical X chromosome that is present in all of the families of these reptiles. So then we decided to analyze the microsex chromosomes in these lizards. So reptiles, they love to have microchromosomes. In this case, they have plenty of microchromosomes. So we, at the end, after a lot of work, we found out that we have 1 microchromosome. In this case, it's the orthologous microchromosome to chicken's chromosome 17. And then this is what we kind of like the pattern that we expect when we have differentiated sex chromosomes. So in females here, we have this coverage at the genomic level, and then in males, we have exactly half of the coverage that we see in females, meaning we only have 1 X chromosome in males, and when we have 2 X chromosomes in females. So somehow, this is very strange, we used to have an XY system, and then we changed completely, but we changed to have a new XY system, which is completely strange, right? And then it's also highly degenerated. We only found like 13 genes on this Y chromosome. And then we see, when you actually see very few genes on your chromosome, you kind of think like this chromosome should be a little bit old. So so the idea was, because these chromosomes stop recombination, you can actually use mutation rates to date when are these chromosomes originating. And then we realized that these chromosomes originated around 68 million years ago. So what happened 68 million years ago that could have triggered the change of a XY system to a completely different, new, novel XY system? So let me tell you a reminder that basilisk, the whole family of Corytophanidae is found mostly in Mexico and Central America, a little bit also on the north of South America. And then what happened 68 million years ago exactly at this location? So what happens is we have a massive meteorite impact in the Yucatan Peninsula. So which is very strange is that, so this is how it looked like Mexico back in those days. And then the close families to Corytophanidae, which are Crotaphytidae, Phrynosomatidae, they have the old XY system, and they're also located exclusively in Mexico and United States, right? So they're mostly on the mountains of Mexico and United States, on the northern part of Mexico. The Corytophanidae are actually very close to this location, changed the system. So somehow we actually, this is going to be very difficult to show, but it kind of like, there is an overlap between the change of the sex system that is a very conservative system. It's found in all other different families, and it's been there for 160 million years. And then suddenly we have this, this meteorite coming, this asteroid coming, changing a little bit exactly where these lizards are living. And then somehow that changes, that's what we think, it changes the whole population of this lizard and then made possible a switch of a new XY system. The details of this are going to take a little bit longer. We need to probably sequence many more of the genomes and see what's happening more at the population level of these lizards. And then with this, that's the end of the talk. So these are the different organisms that are working specifically on transitions of sex chromosomes. So these are the collaborators that work in Mexico and abroad on the different subjects. And this is the list of people that have been working on these specific subjects in the lab. So, and I thank you very much for your attention.
Thank you very much. That was a fantastic talk. Is there any questions? We have plenty of room for them. Don't be shy. Yeah. As you wish.
Sí, o sea, yo creo que no sé, o sea, hay dos sobrelapes muy interesantes, y es el momento en que aparece la familia Corythophanidae, también el momento de especiación de la familia también coincide con este mismo lapso, ¿no? En el momento en que aparece el sistema sexual y que está este evento. Ahora, yeah, all right, sorry. So she asked about if the location, geographical location close to the impact of the asteroid had a role in changing the sex system. So what we see normally, that's something that we have seen in different systems, and then that you have a relationship, like a correlation between speciation and the origin of new sex systems. So the moment a lineage has a new sex system, normally it comes also with the speciation, which makes sense because if you have a new sex-determining gene, you can now go back to the old, so it will be like a very chaotic system to have two sex-determining genes. So that really helps to have speciation. And then in this particular case, we have the origin of the Corythophanidae family almost at the same time that you have the origin of the sex system, which is not rare, but also you have this impact. So the other families that are also only located in Mexico, the Phrynosomatidae and Crotaphytidae, that are a little bit further away, they remain with the old, very conserved, extra, already proved, let's say, sex system. And it's only the Corythophanidae that are located in this particular region, that is South Mexico and Central America, close to the impact zone. So what we think somehow is that, in general terms, in like the evolution of sex systems, is that to change one sex system from a very conserved that is really working, no, that is you have a strong selection on this system, to a new one, is that you need to decrease your population. So you need to go to a very low effective population, and then by drift, you get, because you have determinant genes appearing all the time, right? But they don't get fixed because the ancestral system is on the very strong selection. So if you go to a very low population, then you have drift, and then in that case, you can actually switch sex system. So this is what we've seen in some rodents around the world. So rodents that live in very small islands, and they have very small population, they tend to have weird XY, our system, but really weird. And some of them actually lost the Y chromosome. But it only has happened in very small populations. There is something about having a small population that makes, because our system is very old as well. It has 180 million years. So it's very difficult to change a system that is being there for, is so under selection. So you need to go to drift. So that is what I think it happened. So you went to drift, So the impact made the whole population go to a very low numbers, and then it caused a new sex system, and with the new sex system, it caused speciation. That's a very, very good question. I think sorry. So he was asking about segmental duplications, because normally sex chromosome, specifically the Y chromosome, has a lot of segmental duplications. And in our case, 90% of the Y chromosome in mammals is actually made of segmental duplications. And in rodents, it goes up to 99%. So he was asking whether we see the same in other model organisms like lizards. So the problem here, I think we will see eventually that they have lots of segmental duplication, but it goes together with stop of recombination. The problem is that we need to have the full sequence of the sex chromosomes. And then sequencing the Y chromosome is very, very tricky because it has lots of segmental duplications, and because the problem is that if we take DNA and then we sequence, because the Y chromosome represents like 0.1% of the whole genome, you get very, very few reads, even if you sequence like crazy. So you need to have very specific methodologies that take out, they capture the Y chromosome, and then you sequence like crazy. So normally what people do, there is one lab actually in the world that is doing this. It's in the United States. It's David Page at the MIT. So they have a special system that they capture the Y chromosome, and then they put it in a bike library. And they sequence for 10 years the bike library to get the segments. So that's why we got the Y chromosome in humans. That's why we got the Y chromosome in mouse. So there are like 5 Y chromosomes that we know. And all of them have different structures for segmental duplication. So it will be very, very interesting to see what's happening in different organisms. But it's going to take time, probably, to improve the technology of sequencing like with like big depths and then long sequences at the same time. Yeah. Anybody else? Yes.
Your example with the novice was a specific gene that you were able to follow and see the results. Is that happening in the other reptile species, or you know?
About you mean like the compensation mechanism, the balancing? So far, anolis is the only case that we have fully studied in detail, because it's the only reptile, it's the only lizard with a full genome sequence. So we do have a big problem in reptiles, that is we don't have enough genomes. So anolis is the model organism because we have the reference genome. I think that we have very few solutions for the problem. So you either inactivate one X chromosome in females, so you either upregulate one X chromosome in males, if you want to have full balance. So what we're actually seeing in Coleoptofanidae, that they also have the XY system, is that you only have partial balance. That is, you only upregulating certain genes. So the idea here is that you need to balance those genes that will be lethal to have in only one copy. All right? It really depends, it's completely random. If your original chromosome that became a sex chromosome had a lot of these genes that we call haploinsufficient, then you need to have a full chromosome Y level rebalanced. So this is what happened in our case. This is what happened in marsupials as well, and this is what happened also in lizard. But if your chromosome, pre-sex chromosome, have like 2 or 3 genes that are haploinsufficient, then you don't need to have a whole full system that for the Y, for the entire chromosome. You just need to restore balance of these genes. And it's actually, if you have very few genes, it's very easy to restore balance if you take those genes out of your sex chromosome and put it on an autosome, by retrotransposition or by translocation. You have very few genes, you just move them to an autosome, and that's fixed. That's something we've seen in other systems. Mm-hmm. All right, well, thank you very much. Andres.
Sorry, guys, we only have one microphone, so we have maybe to share it. So Diego, I have maybe a more basic question about the XY system. How flexible it is, or how can it be measured, the flexibility of the system? Because if it's in charge of sex determination, it seems to be pretty critical for species in general, but at the same time has been tolerating the gene loss of big chunks of DNA. So I don't know, is there a way to measure this flexibility, or how conserved it will be for, I don't know, future generations in different species? Is there a way to model that, to know how flexible the system can be?
Well, thank you. Well, it seems the system these systems are not very flexible. And that's, specifically in mammals, it's very difficult. I mean, in reptiles, we think that reptiles can also have temperature sex determination. So we think that in reptiles, it's easier to change from one sex system to another sex system, because you can always go back a little bit for a while to temperature sex determination, and then gain again sex chromosomes. So this is something that we try to understand. But in other organisms like mammals or like birds, that the embryo, the temperature of the embryo is constant, it's very difficult to change to a different system. So we will need to change immediately from a sex system to a different sex system. So I was telling you about these rodents that live they have very small populations, lots of drift, and then they have these weird sex chromosomes, and they have lost the Y chromosome. So actually, in all of the cases that we've known so far, they completely lost the Y chromosome, but they didn't lose all of the genes from the Y chromosome. That is, many of the genes that used to be on the Y chromosome jumped out of the Y chromosome to an autosome, and that autosome is now the sex chromosome. So let's say they started all over again with a fully new, non-degenerated sex chromosome, but you're actually carrying the same genes. So you are not fully escaping the ancestral sex system. You're just restarting with a full new chromosome. So this is a specific case of mammals. In fish, amphibians, and reptiles, we do see lots of changes from one system to another system, and it can be completely different chromosomes all the time. So it's more dynamic. And I think because they have these alternative sex systems, like environmental sex system or social. I mean, fish, they have lots of social sex determination that we don't have. So in fish, it depends of many factors. Socially speaking, it defines which fish we're going to be males and which one going to be females. Although genetically you are male, but the group will decide whether you stay as a male or you will become a female. So, and because we don't have that, then in our specific case, we are fixed. So what we've seen is that we can actually model is how fast this system decayed. And then we've realized that it goes very fast. In the first million years, you've lost almost 90% of the Y chromosome, and then it stayed in stasis with the same amount of genes for very long periods of time. So it can stay like that for many, many, many million years again.
So I know that in mammals, at least in humans, there are some sex chromosomes abnormalities like Turner syndrome and these kind of things. So I was wondering if something similar has been seen in reptiles too, and if that can tell you something about how things are regulated or not. Because I mean, Turner, they can be normal people to a point, so.
So the Turner syndrome is when you have only one X chromosome. So these are females that only have one X chromosome, and these 2% of genes that I've been telling you about, they are very important for development. And then the Turner syndrome, there are these people that are very short and then have lots of different cardiovascular problems, and they're infertile and things like that. So you can actually correct a little bit of the Turner syndrome with the hormones if you detect it like in the first weeks after birth, and you give an hormone therapy, then you can restore like almost full development, normal development. Do we see similar syndromes in other species? It's also, again, it depends on these haploinsufficient genes. So Turner syndrome happens because our sex chromosomes are enriched in haploinsufficient genes. So when you have, even if you lack these 2% of the genes, these 2% are highly haploinsufficient. That's why they were kept on the Y chromosome. If they are not there on the Y or the second X chromosome escaping inactivation, then you only have one copy, and that's bad. And that's why we have this Turner syndrome. So it will depend on how many haploinsufficient genes you have on your sex chromosomes. So nobody has actually realized that whether we have or not an equivalent of the Turner syndrome in reptiles. But now that we actually located the sex chromosome, let's say in Corytophanidae or in Anolis, in different species, then we can actually do assays at the population level and see how many times do we see reptiles missing one of the sex chromosomes. And then when we have this number, we can actually ask questions whether these specific individuals look different or not. But we are not there yet.
So thank you very much. I think he deserves a round of applause, please.